Bismillahirrahmanirrahim. Assalamu alaikum. I welcome you all in the third and the last day of the training course. Today we have two virologists as the resource persons, that is Dr. Hana Iqbal and Dr. Irfan Khan. They will be discussing today PCR technicalities and data interpretation. I request now Dr. Hana Iqbal to start the session.
Good morning, good afternoon, and good evening. Assalamu alaikum. Let's start the session. Bismillahirrahmanirrahim. Okay, we are having some. So today I'm going to discuss with you the real-time PCR for COVID-19 diagnosis. So, we are having some technical difficulties. Please be patient. Okay, so we will be discussing real-time PCR for COVID-19 diagnosis. So let's just first solve the problem. I think most of the people cannot see the screen I'm sharing. Okay, so once again, real-time PCR for COVID-19 diagnosis. I'm Dr. Hana Iqbal. I have been associated with Dr. Pandwani's Center for Molecular Medicine and Drug Research, International Center for Chemical Sciences and Chemical and Biological Sciences since 2007. So first, what is polymerase chain reaction? Polymerase chain reaction is an amplification technique for cloning the specific or targeted parts of a DNA sequence to generate thousands to millions of copies of DNA of interest. And there is another extension to PCR, that is the real-time PCR, which detects the products during the linear phase of amplification. And the amplified copies are actually detected at the time of generation rather than at the end of the cycle, when the cycle is completed. This technique is based on the probes that fluoresce only when they are incorporated into the PCR product. So how real-time PCR is different to conventional PCR. In real-time PCR, the amplified DNA is labeled usually with the fluorescent dyes, which enables the amplification of DNA to be visually detected. While conventional PCR can only be visualized when you run it on the through electrophoresis. In real time, the fluorescence is measured after each cycle, which enables the testing to be read, analyzed in real time, as compared to the conventional PCR, which can also be referred to as the endpoint PCR, in which the result is only read at the end of the cycle. The steps for annealing and extension during replication of DNA is usually combined during qPCR testing. Annealing and extension are done separately in conventional PCR. So to carry out PCR, especially when you are dealing with viral samples, you have to have a specific BSL-2 laboratory setup. For that, clean laboratory setup. Let's see what we have here at NIV to do the PCR for diagnostic purposes. for PCR, for diagnostic purposes, the lab should be well equipped and it should be a clean lab. It should have a PCR cabinet. It should have a PCR cabinet, which is a clean cabinet, which has a certain flow, which protects the sample and all the reagents that we are using. Okay? It should also be equipped with exclusive cold storage devices that is dedicated for only storing for the kit and related reagents. No samples, RNA should be stored in it. The laboratory should also be equipped with small equipments, including micropipettes and mini centrifuges and different supplies, including tubes, tips, etc. The tips should be filtered. Before starting, clean the PCR cabinet with 70% ethanol. Also clean all the mini equipments that will be used during the process, and turn off close the sash of the PCR cabinet. So here we are cleaning it with the 70% ethanol. We also clean all the small equipment as well, including the micropipettes. Close the sash of the cabinet and turn on the UV light for 5 to 10 minutes. So for storing, we use exclusive, or you can say dedicated 4° and -20° freezers for storing the PCR kits and reagents. Okay. There are several kits available in the market for diagnosing the COVID-19 samples. Currently we are using GeneSick real-time PCR. Currently, we are using GeneSick real-time PCR coronavirus COVID-19 by Primer Design. This kit is specifically intended to be used only to detect SARS-CoV-2 in a qualitative manner, and it detects the viral RNA, which has been extracted from the nasopharyngeal swabs, oropharyngeal swabs, or sputum, and it is IVD system. It is used with the IVD system, extraction system, which is the in vitro diagnostic system. Okay, now Kasali will take over for translating this slide into French.
Ok, merci beaucoup pour avoir participé à la dernière session de notre formation. Au cours de cette session, nous allons avoir des intervenants. Et la première intervenante a juste commencé son speech, son discours. Alors, il s'agit de Dr Anna Iqbal, comme elle s'est présentée. Donc, la première thématique porte sur le temps réel de la PCR pour le diagnostic de la COVID-19. Alors, on a évoqué la chaîne de réaction de la polymérase. Donc, la chaîne de réaction de la polymérase, c'est une technique d'amplification pour le clonage de la cible. Donc, le temps réel de PCR, le temps réel de la PCR est différent en fait du temps conventionnel de la PCR. Alors, une grande différence se trouve au niveau de l'amplification, avec le temps réel de la PCR permet d'avoir une application plus forte pour l'amplification de la DN, donc de la DNA, et pour faciliter une bonne lecture. Alors, en ce qui concerne l'équipement utilisé comme PCR dans notre labo de biosécurité du niveau D, comme vous allez voir dans la vidéo qui a été présentée, et dont le sous-titrage a été en français. Donc, l'appareil comprend une assiette PCR, ça comprend des micropipettes, vous avez une microcentrifugeuse, vous avez des boîtes de pointe, vous avez aussi un congélateur. Alors, avant, il faut d'abord nettoyer l'appareil. Il faut nettoyer l'appareil et tout l'équipement de la PCR avant de pouvoir commencer la manipulation. Nous avons encore parlé de de la PCR, donc de la PCR temps réel et PCR COVID-19 spécifiquement, nous avons un modèle génétique qui permet de pouvoir faire une analyse qualitative de la détection de SARS-CoV-2 dans l'échantillon de l'ARN viral prélevé dans les différents échantillons au niveau avec l'écouvillon, au niveau de la cavité nasopharyngienne, au niveau de la cavité oropharyngienne, mais au niveau aussi des autres échantillons biologiques. Merci beaucoup.
Thank you, Asari. So now proceeding further. So this kit, GeneSeq Real-Time PCR Coronavirus COVID-19 kit consists of OSEG RT-qPCR master mix, which is like in lyophilized form, 2 vials of this COVID-19 primer and probe mix, 2 vials, OSEG resuspension buffer, 2 vials, template preparation buffer, 2 vials, water which is RNase DNase free, 1 vial, COVID-19 positive control template, 1 vial, and GeneSeq Easy RNA internal extraction control, 2 vials. I will be explaining why we will be using controls, positive, negative controls, and further. Okay. So here is an overview for the PCR kit, which we are currently using, GeneSeq. This is the master mix lyophilized form. This in the amber colored tube, we have the primer probe mix, which includes the probe mix for internal extraction control as well. Then blue cap, we have 2 types of blue capped tubes. One is for internal extraction control, and the other one is for the OSAG resuspension buffer. So you should never be confused between these, and while using this, we have to be careful which tube we are using actually. Yellow capped tubes are for template preparation buffer. I will be explaining their use as well later on. The white capped is the RNase DNase free water, which we use as negative control or NTC. And then we have rat capped, and then we have rat capped tube, which is the positive control. Okay. So what is the purpose of primer and probe mix? The GeneSeq COVID-19 assay includes a primer and probe mix which contains a SARS-CoV-2 specific probe that is labeled with the FAM-fluorophore. The oligonucleotide primers and probe for the detection of SARS-CoV-2 are from the ORF1AB genome region. Primers and probes to amplify and detect the internal extraction control RNA template is also included in the primers and probe mix. The IEC, that is the internal extraction control specific probe, is labeled with the hex fluorophore. The other tube is RNA internal extraction control. The GeneSeq Easy RNA internal extraction control template is added to the nucleic acid extraction system. That is when you are extracting the RNA from the nasopharyngeal swabs. It provides an RNA template control for the extraction quality and efficiency. It also detects the PCR inhibition, if there is any PCR inhibition due to any kind of reagent or anything, and it confirms the integrity of the PCR run. This reagent should be handled with caution in a dedicated nucleic acid handling area to prevent possible contamination. The GeneSeq Easy RNA internal extraction control template is not related to the SARS-CoV-2 viral sequence. GeneSeq COVID-19 positive control. The GeneSeq COVID-19 positive control template, which is also known as PCT, is provided in a sealed foil envelope and contains a high copy number of template. It is provided into a sealed foil envelope as to prevent any contamination with the other kit reagents, because if it is contamination, it will result into false positive results. It should be opened and processed carefully to avoid cross-contamination, because the point I have just raised. The PCT tube contains synthetic DNA, which represents the SARS-CoV-2 genomic region of interest. Following the resuspension of the PCT, the concentration of the PCT in the vial will be 1.7 into 10 raised to power 5 copies per microliter, which is a very high concentration actually. That's why you have to be careful. To ensure it ensures the PCR run validity. The PCT should it should produce amplification in the FAM channel. Then we have negative extraction control in the kit, which is also known as NEC. So when you are setting up your experiment for RNA extraction, at least one negative extraction control is prepared each time. The NEC is an extraction control with no clinical specimen or sample added to it. It is prepared by extracting from RNase, DNase free water, which is provided with the kit. The IEC, or the internal extraction control, is added to the NEC sample during extraction, which is directed by the manufacturer's instructions. This NEC will serve as the negative control for the entire testing system and to check for the contamination during the PCR plate setup. Apart from positive control and the NEC, that is the negative extraction control, we have no template control, which is known as NTC, which we use as when we use the DNase RNase free water and during PCR. The NTC is used to check for the contamination of any possible contamination during PCR plate setup. So first we need to pre-suspend 4 components in the which is provided with the kit, because these are provided as lyophilized products. One is the OSAC PCR master mix. The second one is COVID-19 primer and probe mix. Third is the positive control template, and the last one is the RNA internal extraction control. So, okay, let's see a video about how to resuspend these. So prior to preparation, reagents should be allowed to talk properly, and all the calculation must be completed based on the number of samples. For example, when you are preparing for 1 sample, the required volume for the OSAC qPCR master mix is 10 microliter. And for the same number, for 1 sample, the primary and probe mix should be 2 microliter of this should be added and mixed together. So if you have more than 1 sample, for example, you have 10 samples, then you will be multiplying 10 with 10, which will be 100 microliters for 10 samples. That is for the qPCR master mix. And for the primary and probe mix, that would be 20 microliter. You will add both of these into 1 tube and then take 12 microliter of this and add to 1 well for 1 sample. So let's proceed with the video. To prepare qPCR mix, add 5 to 5 microliter of OSAC resuspension buffer in each vial of the qPCR master mix and mix it gently. Sorry, I'm continuously clicking wrong buttons, so I'm really sorry. So this is how you prepare the PCR master mix. And then you have to prepare both the vials. Both the vials constitute for 96 samples actually. The whole kit is for 96 samples. You have to be careful, you have to be gentle while mixing it as to prevent any bubble formation or anything. Then for primary and probe mix, add 110 microliter of template preparation buffer, that is yellow cap tube. Mix it gently, be careful because this is sensitive to light. So you have to be careful, and please don't pipette it harshly because it can result into degradation of the probe. So mix it gently. There are 2 amber colored tubes for primer probe mix. Then prepare internal extraction control by adding 1000 microliter of template preparation buffer, which is the yellow cap tube. Mix it gently. Now we have taken the buffer and we are adding it to the IEC tube, mixing it. You have to be gentle because otherwise it will make bubbles in the tube and that can interfere with your process. Now take 600 microliters of template preparation buffer and add it to the positive control tube. You have to be careful while making the PCT because it can result in cross-contamination. And it has, as mentioned earlier, it has high template concentration. So we have to be really careful. Discard the tip really carefully without passing from top of any of the tubes or anything. You have to be really careful in handling this and be very gentle and slow. You don't have to resuspend the negative control because it's already RNase, DNase free. Now to prepare the master mix, just label a tube with date, master mix initial, and then you can also label it with the kit name as well. I will be showing you how to make a master mix for 96 samples. So for 96, we have to take the whole of the content of the qPCR mix that is in the glass vial, as you can see. We have to be careful while mixing it through pipetting. Take out and put it in a fresh tube from both the vials for 96 samples. So for this setup, we use 96 samples, including the PCT and the negative control as well. Now gently, when you have added the master mix, now you have to add the primer probe mix. Be careful with it because it's light sensitive. 110 microliter of 110 plus 110 actually, 220 microliter of primer probe mix will be added to the whole qPCR master mix. It is recommended that before taking the primer probe mix, gently centrifuge it, short spin actually, because when you are preparing the primer probe mix, there will be some which will be on the cap or on the walls of the tube. So just spin it to settle all the drops down, and you can then pick up the whole volume actually. Otherwise, you can miss some of the volume. While you are preparing any reagent or while you are using any reagent, immediately cap your tubes to prevent any kind of contamination or spill. It's true with all kinds of tubes and reagents. Now, spin it for a few seconds, like 10 to 15 seconds, to ensure proper mixing and settling of the master mix. Now take 12 microliter of the prepared master mix and dispense it in PCR strips or plate, whatever you are using. You always use PCR cooler during and beyond this step because this mixture is sensitive to temperature and light. So you have to be careful with temperature. And while you are pipetting for 96 samples, it will take some time. So the temperature of the master mix will increase, and which can actually hinder with your process. So always use PCR cooler. So we have dispensed the master mix in the whole of the 96 wells now. Make sure that all the wells they have the master mix. None of the wells have been skipped. Okay. So over to Kasali for a brief French translation.
Ok, merci beaucoup pour la parole accordée. Alors, on avait commencé avec le processus d'extraction du témoin. Nous avons expliqué que c'était pour amplifier. Alors maintenant, on va parler du modèle sans témoin. Donc, la récipiendaire a parlé du modèle sans témoin. Donc, le modèle sans témoin est utilisé pour vérifier toutes les contaminations durant le processus de PCR. Alors, par rapport maintenant à la suspension, nous avons, elle a fait allusion au kit de suspension qui comprend les composantes suivantes. Vous avez l'OASIC, donc qui a un mélange mètre. Vous avez les amorces. Vous avez les amorces et la sonde. Donc, le mélange amorce-sonde de la COVID-19. Vous avez le contrôle positif, donc les témoins positifs. Donc, le modèle est fait par les témoins positifs. Et vous avez aussi un modèle d'extraction, donc un motif du témoin négatif. Alors, en ce qui concerne le mélange, la préparation du mélange mètre, nous avons une réaction pour démarrer la réaction. Donc, pour préparer un mélange mètre, donc les différents réactifs sont utilisés. Normalement, pour la resuspension des composantes, vous avez l'OASIC qui est utilisé à un volume, donc le volume recommandé est de 10 microlitres. Vous avez aussi le COVID et vous avez aussi les amorces et les approbes, donc avec un volume de 2 microlitres. Alors, il faut ajouter 12 millilitres dans chaque membre comme il est recommandé pour tester avec pour tester la plaque appropriée, pour choisir la plateforme du PCR en incluant comme le CPT et aussi comme le NEC. Donc, c'est un médecin qui peut être préparé à partir d'une composante NEC. Alors maintenant, en ce qui concerne la resuspension, comme la vidéo a expliqué, d'abord il faut préparer le mélange PCR en ajoutant 525 microlitres du tampon de suspension OASIC dans chaque flacon et mélanger délicatement. Pour le mélange, ensuite, pour le mélange des amorphes et de la sonde, il faut ajouter 110 microlitres de tampons de préparation dans la matrice. Ensuite, il faudra préparer les témoins d'extraction interne en ajoutant 1000 microlitres de tampons de préparation du modèle, donc un modèle qui est considéré comme la matrice ici. Ensuite, il faudra préparer les témoins modèles positifs en ajoutant 600 microlitres de tampons de préparation de la matrice, bien du modèle. Ensuite, il faudra mélanger les PCR et mélanger les amorces sondes dans un tube et centrifuger pendant 10 secondes pour assurer un mélange correct. Alors enfin, maintenant, il faut boucher correctement les bandes et les transférer sur la matrice. Merci.
Thank you, Kasali, for the nice translation. Okay, let's move forward. So the next step is RNA addition into the PCR master mix. So the reaction step is set up according to this kit you will be adding 8 microliter of the following into the appropriate wells according to your plate setup. RNA samples, it depends how many RNA samples you have. The second one would be the COVID-19 positive control template. It is usually recommended, and it is our practice as well, that we use, we add the positive control at the last, in the last well, to prevent any kind of contamination. Then NEC, that is the negative extraction control, and NTC, which is optional, but we usually use it. So seal that plate or the tube strips with an appropriate seal or caps and place in the RT-PCR thermal cycler. So let's see how it's done. So first requirement is you have to wear double gloves, because now you are dealing with viral RNA samples. You have to be careful, and actually it serves when, if you by any chance get any kind of contamination or sample on your hands or on your gloves, you can take off your outer pair of gloves in the safety cabinet and change and wear a fresh one, because it is not recommended to take out your hands wearing gloves which is contaminated. So now they are taking out the RNA from the -20 freezer, because if you have some time before using the RNA for like 5, 10, 20 minutes, maybe you can put it in a fridge or a freezer. So now this is a plate which was prepared earlier, and all the well contains master mix, PCR master mix. So remember, 8 microliter of RNA template will be added in their corresponding wells. The white plate contains the RNA sample freshly extracted, and it also contains the NEC, that is the negative extraction control. Be very careful while adding the RNA samples into the well. The tip should not touch any adjacent or neighboring wells or the walls or the plate. And also change tips with every sample. The tips used should be filter tips or the barrier tips. You should be slow and gentle while performing this step, as to prevent any kind of contamination or accident or spill. Because this, as you have seen yesterday, this RNA is extracted on the basis of the magnetic beads. Sometimes we actually face a problem that some magnetic beads are present in the well. So what we do, we actually spin that sample and take the supernatant and use it for the PCR. So, and any accident can happen while you are, pipetting the RNA. So like here, the worker actually touched her tip with her glove. So first thing would be to cover her plate, because she needs to protect her samples, especially the PCR plate. Now she will be adding, she will be spraying her gloves with the alcohol, 70% alcohol. She's taking her outer pair. This is what I was talking about, because in the safety cabinet, her hands should not be bare. So there should be an inner pair as well. So now she is wearing a fresh pair of gloves to proceed with her test. Okay, now she will be proceeding with her plate. Now we are adding the last few samples into the master mix. We leave few of the last samples for the controls, a few of the last well for the control. The last few well in the RNA plate, the white plate, are already NEC, that is the negative extraction control. So we have added that to the PCR plate. Now we will be adding the negative control, that is NTC, no template control, and PCT, which is the positive template control. This is the PCT. You have to be very careful while you are adding this, because it can lead into a massive contamination and can result into false positives. Be very careful and immediately cap all your tubes and place it away from your plate. You have to be really careful. So this is the negative control. Cap it, place it away in the rack. Now we have covered the strips with the caps, and now we are doing mini spin to settle all the reagents into the well. You should check for the volume in each well. It should be equal. It should be 20 microliters because you have added 12 microliters of master mix and 8 microliters of RNA. That means your whole volume should be 20 microliters. So you have to be careful that it is 20 microliters. If you see that any of the well is not according to the other wells, or you should have an idea that it should be 20 microliters. If it is not, then remake that well or the whole strip, which is more convenient. Okay. So first let's have a French translation for this step. Are you ready, Kasali?
Okay.
Okay. Over to Kasali.
Okay. Merci beaucoup encore pour la parole. On venait d'aborder le point sur l'addition de l'ARN. Donc, il faut d'abord démarrer la réaction. Et en démarrant la réaction, il faut ajouter 8 microlitres de chaque composante suivant. Donc, vous avez la première composante, c'est l'ARN, donc l'échantillon de l'ARN. Vous avez la matrice du composante positif, donc le PCT. Vous avez ici la matrice du contrôle positif, témoin positif. Vous avez le contrôle négatif. Et puis maintenant, vous avez le NTC qui est optionnel. Donc, ce n'est pas obligatoire, c'est optionnel. Alors, vous allez... donc, pour maintenant, comme procédure, il faut initialement porter les gants. Puisque nous sommes dans de manipuler les virus, comme nous manipulons les virus, la première précaution, il faut porter les gants. Alors, pour préparer l'échantillon, donc l'ARN, donc pour pouvoir préparer l'ARN dans chaque matrice, donc c'est l'ARN qu'on va utiliser, c'est l'ARN qui est fraîchement extrait, puisque qui vient directement de l'échantillon. Donc, il faut prendre les plaques préparées au préalable. Il faut ajouter à l'échantillon dans le puits de la plaque. Il faut le faire lentement pour éviter toute contamination ou bien tout accident. Donc, il faut le faire lentement. Ensuite, il faudra couvrir les plaques pour les prélever, spécialement pour les PCR. Il faut encore laver une fois les mains. Il faut porter une seconde. Donc, pour la deuxième fois, il faut porter un jet de gants. Il faut ajouter les derniers dans le mélange maître. Donc, il faut ajouter les témoins négatifs. Par la suite, il faut encore ajouter les témoins positifs. Il faut couvrir tous les tubes. Donc, il faut être prudent pour éviter toute contamination. Maintenant, il faut vérifier les volumes. Le volume doit être de 20 microlitres. Donc, de 20 microlitres. Et il faut être très prudent. OK, merci beaucoup.
Thank you, Dr. Kasali. Okay. So I hope you have understood till here. And now I will briefly explain what will be the amplification program, because the details will be provided in the next segment by Dr. Irfan, because he will be taking over after me for the next software setting and all the program and everything. So for the reverse transcription step, the time will be 10 minutes at 55 degrees centigrade, and the cycle will be 1. For initial denaturation, it will be 2 minutes at 95 degrees centigrade, cycle will be 1. For denaturation, 10 seconds, 95 degrees centigrade, annealing and extension for 60 seconds, 60 degrees centigrade. And both the last steps, denaturation, annealing and extension, will be for 45 cycles. The channels that will be used will be FAM and WIC or HAX because they both are similar. So while you are proceeding with the PCR for diagnostic purposes, you can always encounter false negative or false positive results. The reasons for false negative can be many, including unsuitable collection, handling, or logistics of the samples. It can be the collection of the samples is outside of the viremic phase, failure to follow manufacturer's kit or the procedures, use of unauthorized extraction kit of PCR platform, that is the thermal cycler, and the reason for false positive results may be due to unsuitable handling of samples that contains the high concentration of COVID-19 viral RNA or unsuitable handling of positive control templates. So that's why I have been emphasizing that you should be really careful with handling the positive control. And also, of course, the positive samples as well, because you never know which sample is positive sample. So you have to be really careful while handling all the samples. And then one of the reason for false positive result can be unsuitable handling of amplified product, that is the PCR product as well. So you have to be careful while storing it and everything. So there are chances of contamination during this whole process. So how we can prevent any kind of contamination during PCR, especially during diagnostics. So the positive, as I have mentioned and emphasized earlier, the positive control template can be one of the major source of contamination, and it should be handled really carefully. It should be opened and processed carefully away from the test samples and other kit components to avoid any cross-contamination. Also maintain separate areas for handling of the specimen preparation, PCR setup, RNA pipetting setup, and post-PCR amplified nucleic acids. Maintain separated dedicated equipments, including pipettes, microcentrifuges, and supplies that includes the tubes, pipette tips for handling of the specimen preparation, pre-PCR assay setup, and post-PCR amplified nucleic acids. Always wear a clean lab coat and disposable gloves when setting up this assays. You have, as shown earlier, change gloves regularly, and whenever contamination is suspected, you have to change the gloves. Keep reagent and reaction tubes capped or covered as much as possible. Always check the expiration date prior to use. Do not use expired reagent. Also, the manufacturer has mentioned it that you should not substitute or mix reagents from different kit lots, even the lots, or from other kits or manufactured by other manufacturers. Change aerosol barrier tips between all manual liquid transfer. During preparation of samples, compliance with good laboratory technique is essential to minimize the risk of cross-contamination between samples and the inadvertent introduction of nucleases into samples during and after the extraction procedure. So the next, okay. So the another few more steps which can be taken to prevent contamination would be when mixing reagents by pipetting up and down. This should be done with a volume roughly equal to 50% of the total component volume. That is, if you are mixing 1000 microliters of the volume, you should set your pipette at 500 or 600 microliters. Work surfaces, pipettes, and centrifuges should be cleaned and decontaminated with cleaning products. That is, you can use 10% bleach, 70% ethanol, or DNA RNA remover, that is also known as DNA RNases. Okay, to minimize risk of nucleic acid contamination. After each run has been set up and performed, clean work surfaces and equipment with a DNA RNA remover. Handle post-amplification plates with care to ensure that the seal is not broken, as to prevent any cross-contamination. Okay, Use RNAs, DNase-free disposable plasticware, and pipettes reserved for RNA-DNA work, that is dedicated pipettes, to prevent cross-contamination with RNases, DNases from shared equipment. Use DNase, RNase-free filter tapes throughout procedure to prevent aerosol and liquid contamination. So now let me just summarize the whole procedure which I discussed today. First, you have the kit, you have to resuspend few of the components that include the primer probe mix. You have to resuspend it in template preparation buffer and store at -20 till further use. Resuspend one step OSAG master mix in with 525 microliter of OSAG resuspension buffer and store at -20 degrees centigrade before using. Also resuspend RNA IEC, that is internal extraction control, with 1000 microliter and PTC in 600 microliter of template preparation buffer and store at -20 degrees centigrade. When you start with the RNA extraction using the nasopharyngeal swabs, add 20 microliter of resuspended IEC, that is the internal extraction control. You have to be careful that you don't forget this. Into the extraction system, this is buffer. Extract the sample RNA according to whatever platform you are using. Prepare reaction mix by mixing 10 microliter of OSAG master mix and 22 microliter of primer and probe mix. Prepare the RTQ PCR plate. Load reaction mix, that is 12 microliter per well. Add RNA sample or control 8 microliter per well. Run on qPCR instrument. Analyze data and report result. The last two part will be discussed by Dr. Irfan after the Kasali has translated this part for you. Thank you.
Ok, merci beaucoup. L'Iskander avait évoqué, après avoir expliqué, il a évoqué les programmes d'amplification. Et en ce qui concerne les programmes d'amplification comprend plusieurs étapes. Nous avons par exemple la transcription reverse qui dure 10 minutes. Vous avez la dénaturalisation qui dure 2 minutes. Vous avez par exemple l'extension qui va durer 60 secondes. Alors, elle a ensuite évoqué les problèmes qu'on peut avoir au cours des manipulations. Normalement, les problèmes où on peut avoir soit les faux, vous avez les faux négatifs, vous avez les faux positifs. Et plusieurs raisons ont été évoquées. Par exemple, lors de l'observation des faux négatifs, parmi les raisons évoquées pour pouvoir obtenir les faux négatifs, vous avez par exemple la collection des données, vous avez par exemple les échantillons qui sont, vous allez par exemple, vous allez acheter les réactifs auprès des mauvais fournisseurs. Vous avez par exemple un programme inapproprié que vous allez utiliser pour l'analyse des données. Alors pour les faux positifs, des raisons principales ont été évoquées. Nous avons par exemple l'utilisation d'une méthode inappropriée pour la collection des données, des échantillons plutôt. Vous avez aussi une mauvaise amplification. Alors maintenant, comment il faut prévenir les contaminations? Donc pour prévenir les contaminations, au niveau par exemple du témoin négatif, le témoin négatif est l'une des principales sources pour la contamination. Donc, il faut parfois séparer les zones. Il faut laisser les zones. Les zones doivent être aérées. Donc, il faut maintenir un espace entre les différentes zones. Vous avez aussi l'équipement. Donc, il faut utiliser un équipement approprié pour diminuer les risques de contamination. Vous avez aussi maintenant, par exemple, il faut aussi prendre des précautions au cours de la manipulation. Normalement, il faut changer les gants. Il faut toujours, quand vous utilisez les réactifs, des réactions, il faut toujours bien préciser. Vous devez noter toutes les informations relatives. Vous allez, par exemple, l'une aussi des sources, quand vous allez utiliser un réactif qui est expiré. Donc, si le réactif est expiré, donc la réaction que vous allez mener, toute la manipulation sera faussée. Donc, il faut par exemple, pour les zones où il faut utiliser les aérosols, il faut bien pulvériser de manière appropriée. Alors maintenant, pour pouvoir minimiser aussi ces risques-là, il faut suivre les bonnes pratiques de laboratoire. Donc, le suivi strict des bonnes pratiques de laboratoire est essentiel pour pouvoir délimiter les risques de contamination. Par exemple, quand vous devez manipuler les volumes, il faut toujours utiliser les volumes appropriés. Il ne faut pas aller en deçà ou bien il ne faut pas aller au-dessus du volume qui est indiqué. Alors, pour les surfaces de travail, il faut les décontaminer en utilisant les désinfectants. Par exemple, vous avez l'eau de javel, vous avez l'éthanol. Pratiquement, généralement, l'éthanol est utilisé à 70 degrés, 10 degrés. Voilà un peu en ce qui concerne les préventions contre la contamination. On a encore parlé, elle a en fait parlé de la prévention contre la RNA. Donc, il faut maintenant utiliser, par exemple, un équipement en plastique. Enfin, elle a évoqué les résumés sur la préparation qui a été, toutes les préparations qui ont été faites. Donc, elle a présenté enfin un résumé de tout ce qui a été dit. Je vous remercie.
Okay. So this actually concludes this first segment of today. And I have few questions. I received few questions from the participants. So let's discuss those. So first question is, what are the constituents of master mix PCR? So as this kit is, we are using a commercially available kit for diagnostic purposes. You should keep this in mind. So the master mix PCR is actually prepared by the company, and it is a proprietary product. And we don't know exactly the whole of the components that is in there, but usually it contains the DNTPs, which is required, the enzyme, polymerase enzyme, monovalent, bivalent cations such as magnesium and potassium, and other buffers. And the other question is, what is the role of NTC, that is the negative template control? So NTC, sorry, it is no template control, sorry. So it is used for running the PCR, and it allows to check if there is any contamination in the PCR reagents, or while we are making the plate, is there any cross-contamination from positive control or other positive samples or anything like that. Then there is another question, at which concentration primer probe has been prepared? So keeping in mind that this is a diagnostic setup, and we are using a diagnostic kit, which is in vitro diagnostic kit, also known as IVD. We do not need to optimize the concentration for any of the components, including the primer probe kit, primer probe mix, which we usually do during our research work. So we use the concentration that is recommended by the kit, and we resuspend the kit according to the manufacturer's recommendations. That is, for one vial, which contains primer probe mix for 48 samples, we use 110 microliter of the buffer. So for 96 samples, the total volume of the primer probe mix is 220 microliter. So I hope if there are any more questions, please let me know. Otherwise, we will go to the next segment. Okay, the other question is, which fluorescent dye you use for the fluorescent detection? What is the mechanism for action? Okay, I believe Dr. Irfan will be discussing this as well, but I will give you a short answer for that. The fluorescent detection, the dyes used is for the viral target, it's the FAM, and for the internal control, it is the hex dye. And the mechanism is basically based on the quencher and the dye mechanism. When the dye probe is incorporated, the quencher actually releases the dye or the probe, and it fluorescents and it is detected. What do you think about a mini pooling testing, a combination of five samples? Is it okay to apply? This is a question that needs actually a lot of time to discuss, and everyone has their opinion, and it also depends from where you are getting your samples. The samples are from one area, from one region or whatever. Also, pooling testing has been tested across the world, and there have been mixed results. Some people have said that the pooling testing has helped them detect the transmission and the prevalence of the disease in an area or a region, but some of the people have said that this does not help. It actually depends upon how far your testing is, how strong your testing is. If this testing is to a certain percentage, like 60% of the population, then I think the pooling will help. But if it's not, then I don't think so. Think the pooling will help. Another factor is that if few of the samples are not strongly positive, then they can be lost. Their signal can be lost actually. So there are mixed, opinions about it. Oh, this will be discussed by Dr. Irfan in the next. Yes. Okay. So the question is how high concentration of viral RNA in a sample can be quantified. This will be discussed because this we will be discussing during how to analyze the results. Any more questions? So let me check if we have any more questions. Can we prepare? Can we use serum sample or plasma sample with the same kit? No, this kit is only for the nasopharyngeal and the oropharyngeal and the sputum sample, this gene-seq kit. It is mentioned in the kit. So you before buying a kit or going for a kit, just look what kind of samples can be processed with that kit. So you have to be careful with that, okay? Can we prepare master mix in the application room or require separate room? Application, I don't understand what do you mean by application room? Do you mean the RNA preparation room, or I don't know? But I will answer this question based on my understanding that if you are saying that we can prepare master mix in the RNA pipetting room, no, you cannot. Just separate your room somehow. You don't have to have a separate room. You can, divide your room with a physical barrier, completely divided, okay? So you need a separate room because otherwise there can be cross contamination, and especially in diagnostic, if you get a cross contamination in your system, it's really hard to get rid of it. So be careful. Another question, the type of fluorescent probe and how the quencher get released, any mechanism associated there? I mean the cleavage of bond there. Oh, it's a lot of chemistry involved in there, and it is actually out of the scope of today's topic. It is a long discussion actually. You can contact me on email and we can discuss it. Okay, and we are also short on time. Okay, it's known that coronavirus has more than one strain. Is there a change in the primer sequence between the ORF region is conserved between all the strains actually? That's why this kit is successful, and the manufacturer usually use the target those genes which are conserved between the strains. What is the sensitivity and specificity rate of the RT-PCR? Okay, Dr. Irfan, I guess, will be discussing this with you. Why we are not using electrophoresis in case of RT-PCR? Okay, first of all, if you use the electrophoresis, it's laborious, it's time consuming. Diagnostic does not allow that, and it comes with complications as well. You will be, you can have contaminations, you can have problems. Also, RT-PCR is cost effective, it's rapid, it's not laborious, and it is efficient as well, and specific as well. And it's really sensitive, more sensitive than the electrophoresis or the conventional PCR. Is it possible to carry out direct qPCR from the nasopharyngeal samples? If yes, which kit can be used? I don't know about the kit, but it is possible, and a lot of people are actually researching on it. They are trying to make this kind of kit because it will save you time. So the extraction step will be eliminated and it will save time. So they are working on it. There are kits, but I don't think they are available in the market for diagnostic right now, or maybe they are. Can we prepare master mix in the, we already discussed this. How much the primer concentration used in the type? I have already discussed this. Can we do direct RT-PCR? I have already discussed this. Yes, we can do. Okay, so I think there are no more questions. If any, if I have ignored or missed somebody's question, I'm really sorry. You can contact me on my email or somehow. Okay, thank you. Over to Dr. Irfan for the next segment.
Good afternoon, everybody. It's afternoon time here in Pakistan, whatever is your time. So I will say good morning and good evening as well to all of you. So we are starting. Let me introduce myself a bit. I'm Dr. Irfan Khan. So basically working here is assistant professor in molecular medicine at Dr. Panjwani Center for Molecular Medicine and Drug Research. I'm working here in COVID labs and PCR area. Basically all the steps are involved in the diagnostic. So we work all together, but here is a team and reporting these cases to the government as well. So I will start my presentation with qPCR and qPCR data interpretations. What is the objective of this talk is that how the real-time PCR work, how the real-time PCR setup is basically performed on the machines, which type of instruments or machine we are using for real-time PCR, and how the real-time PCR data look like, how the real-time PCR data is interpreted for the process of COVID diagnosis, and finally we will be talking about some of the troubleshootings and real-time PCR. So here we go to the what is the real-time PCR? I think Dr. Hannah earlier talked about the initial steps of the real-time PCR. That what is the real-time PCR? The name came is the real-time is basically that we can look at the sample behavior and the real time, and we can observe the sample whatever is going inside the PCR reaction, and we can visualize it on the screen. So in the real time we can observe the behavior of PCR reactions. If we look at the summary of the process for this detection system, so the system is basically divided in 3 phases. That is whether the sample collections from the patient, and then the sample is transported, and finally we sort these samples, batches are made, codes are assigned for these sample. Then the next we proceed the sample for the extraction process. The RNA is extracted from the sample, and this RNA can be extracted is either from the with the help of a manual method if there is a very few sample, or the RNA can be extracted by automated machines. So the next step is looking for the PCR setup, that how the RNA is processed for master mix preparations toward the PCR, and finally how the data is interpreted in the PCR. So there is some basic equipments which are required for the qPCR. Dr. Hannah talked about these all setup. I will not go in the fine detail of these setup. So in the PCR reaction, there is some of the basic step that is required in the reaction mixture, and these reaction mixtures basically composed of DNA polymerases, reverse transcriptases, specific primers and probes which are required. The DNTPs are required, which basically adenine, guanine, thymine, and cytosine. The buffers are required, and similarly the template, which is called the RNA, is required. So these all the ingredients or component of a PCR reaction, when mixed together, it composed a PCR reactions which are put in the machines, and then in real time we can visualize these on the machines. Here we talk about a little bit about the target genes which are used for the COVID detections, and this particular reference here, the reference is mentioned. One of the study compared basically the 7 different commercially available kits and the target which they are analyzed. And right now in diagnosis, there is multiple kits are available, and these different companies have a different target region for the COVID detection. Some of the kits are a single target kit that only one gene is used for the detection, for viral detection, and that gene can be ORF1b or N1 or N2, or can be E gene or something else, or there can be a multiple genes, and these multiple genes is always basically with the help of some of the internal control, and most of the cases the internal control which is used is RNase-P. The RNA-P is basically validating the process whether the extraction process is well performed or not. So here the target genes are highlighted. These all are the different viral targets which are used for the detection of COVID-19. Here we perform a PCR setup. That how we perform the PCR setup in this current time. Right now we are performing one of the experiment. I will be showing you this experiment here. This basically the software which we launch over here. The software we launch over here, this basically here this is one of the kit. So this is basically the software. We already prepared the master mix and is kept in the machine. So right now we will be starting the machine. The reaction, the process will be start. So if we look at the program setup, this program is basically composed of steps which are recommended by the particular kits we are used for detection of COVID-19. Here in this particular case, if we look at the step 1, so we can see that this at 55 degrees C for 10 minutes. So this step is basically performed for the reverse transcriptions of the process where RNA, the viral RNA is converted into the cDNA molecule. While the next step is, we can see that step 2 is 95 degree for 2 minutes. It basically deactivate the RNA polymerases, and the step basically denature the RNA molecule. While the third and fourth steps are 95 degrees C for 10 seconds and 60 degrees C for 1 minute. So these steps are basically repeated for 45 times. It means that the PCR machine will cycle between step 3 and step 4 for multiple times. Up to 45 times, while on each cycle at 60 degree, the camera indicate that the optical reading will be performed at this particular step. So up to 45 steps, 95 and 60, and between 95 and 60 degree, the machine will read these reactions for 45 times. So the 95 degree basically denature the DNA molecule, while the 60 degree the annealing and extension is performed. The COVID target is basically amplified in this particular step. If we want to modify any of the steps, so we can easily modify these steps. We can increase or decrease the time. But here in this particular case is it's a recommendation from the manufacturer that we use these particular guidelines. So we are using that, and we are not modifying the system. Here we can you can see the sample volume, which is basically mentioned over here, that how much volume we are going to perform for this reaction. The volume over here is the 20 microliter. So the 20 microliter volume is basically used for these reactions. 20 microliter volume is used basically for this reaction. We can insert a step over here anywhere you want to insert a step. We can paste over here, and we can insert the multiple step over here. So right now is the machine is ready. We put a plate on it, so we will be running this program. So I click OK, so the program is selected. Now this program is selected, and we are going to run. So the next step is, after you select the program, we click the next, and here is the plate is coming. So if someone already made a program or made a plate for this particular reaction, you can select an existing plate from any of your location, or you can make a new plate from this create new basically over here, so we can make a new plate. So whatever wells you want, you put your mouse key and you can select the wells all together. How much you need two column or three column or whatever you can perform over here, or you simply can click on the side link and it select all the 96 well plate. The next step is, once you selected the plate, we need to go for the color confirmations or the dyes confirmation. That we need to know that which particular wavelength I'm going to read this optical plate. So here the select fluorophore options give me these options, and if we look it over here, so in this current form we are using this FAM dye and hex dye. So these two color will be selected for this optical reading system. However, the machine have a multiple color, so we can select from a very low range toward a very high range, so up to a 5-channel machines. So the machine can select a 5-channel and again a multiple color or multiple excitation and emission wavelengths are over there, so we can select them all. So I select the OK. Over here we can see that these two color are selected. Now I need to select the sample type. So the sample type is, here we select the unknown sample type, while we need to load the target name. These are the FAM and the hex color. So the FAM is for a COVID. I can label it a COVID over here, and the hex is basically internal control. The internal control is basically used to validate the process of RNA extraction, whether the successful, during the extraction process, the successful RNA extraction is performed or not. That's validated by the IC or internal control. So I select this all is the process, and again the next step is we need to label these each of the well. So for that, for this labeling process, we can go with the two different phenomena or processes, and that can be whether here in this particular well I can label this sample whatever my code is, that particular code we give, and we can select the sample. So here the sample is labeled over here, or this through a 1-by-1 process we can label this whole plate, or we can take alternative of a barcode scanner. So we put the cursor over here and we can scan the whole barcodes from the system. An alternative option is whatever the sample is, you make an Excel sheet, and after making an Excel sheet we paste it over here. How much the sample you are here, just drag your cursor and you will be able to make the samples. The complete samples are the code are made for all of these 92 samples. So the codes are made for all of the 92 sample. If we look it over here, yes, if we look it over here, so all the samples are selected now, and we make these all over here. So we select these all and we copy it. After copying these all, here they give us import a spreadsheet. So if we look at over here, importing the spreadsheet, the columns from the column row, we select the sample. If we currently look at over here, so it's selected like 1 1 1 2 3 and the numerical number which is chronological going up. So we select all the column together, like each column of a PCR plate. Once the column is selected over here, we can, in the sample name, we can just paste our name and we select OK, and you can see over here that these samples are appearing over here. So in the PCR machine, simply there is always a negative control and a positive control sample. We paste these negative and positive control sample over here. Once we select the OK, so the machine is asking me whether I want to save this particular plate. So I select the yes, and we can save this particular plate this particular plate is saved at some of the particular location. Now we select the next one is the plate setup is performed. Now we need to go to start the machine. It asked me to run. So here the machine give me to open, or the software give me the control on the lid of the machine if I want to open or close it. So as we already put the samples and the machine, I'm going to start it, and the machine asked me where I need to save my plate. I give it a particular location, and we can save this machine, this plate on this particular machine. No, the program is started. The program is initializing and program is starting. In the meanwhile, once the program is starting and is running, and the middle time, we will be sharing this process of this whole PCR machine, and I will be sharing you the detail with you for all of this process. Now, once this program is set up here, in my presentation, all of the details are given, how the cycle is made. So the cycle is made over here for PCR reaction, that how which program we are running. The next one is the plate setup. I already showed the plate setup to you, that how the plate setup is performed. Here is the color selection. The color selection is selected from this select fluorophore option, and whatever these channels are available with you, and among each channel the multiple color, the excitation and emission wavelengths are available with you. It's depending on your PCR target, whatever target you want to amplify. If you want to select all of these color, the complete color set, so you can select the complete color set and it will appear on the machine. But it depends on your probe dye or probe excitation and emission wavelength, which you selected for your PCR machines. And a particular for in the process of this probe selection, there is always if there is a multiple target, so we select the excitation and emission wavelength of these probe which are different from the other probe. Like if someone is a green fluorescence, so a green fluorescence, we look for another color like a red fluorescence, and we look for a third color toward the far red fluorescence, or something which is the excitation and emission is completely different from the other, so that these two probe excitation and emission don't overlap with one another. Once they are not overlapping one another, so in this way it gives us the process that multiple color can be selected. Once the fluorophore is selected over here, so then we can label the sample name. We can label the sample name, or we can label the biological target name. We can label whatever the labeling we want. Here is the import spreadsheet options. Okay, so here is the import spreadsheet option. Among the import spreadsheet option, from the column we need to select the column if we are going to paste all the Excel sheet over here. So the columns is basically selected. This column can be selected by just clicking the small arrow for multiple times, so you can select. And here is the sample name, so we can copy the sample name from the Excel sheet and just paste it over here. We select OK. After selecting OK, all the names, all the colors are appearing on this plate. We give the location to the plate, we save the plate, and we proceed for the next step. Once we go, and the next step here is the machine gave us information that I want to start the plate. So once we start the plate, the machine asks us to save the data plate. So we can save the data plate on a particular location, and the cycle is continued or cycle is started. Once the cycle is started, so it gives us a multiple option. The run status, where the machine is running at current position. It highlighted that particular step, and this particular step is basically moving in between all of these selected options. And similarly, in the real-time options, we can And the real-time option, we can observe the peaks or the fluorescence or the positive or the negative sample which are coming. So before completing a cycle, we can observe the samples well, that the run is valid, whether any of the sample is appearing positive or any of the sample which we are interested in is negative. So and that way these options can be used in the real time. In the meanwhile, once the program is running, the program is started, and there is some mistake or there is no labeling, or we want to label this plate later on, here is the plate setup options. So we click on these plate setup options and it asks us for this screen is appearing. Once this screen is appearing, so we can select a fluorophore again, we can label this plate from the beginning, we can import a spreadsheet, and all of these changes which we are making on a program, once it is running on the real time, that can be made. The machine give us the access to have this all process. Here on this particular slide, so we will be going toward the data interpretations. Among the data interpretation options, if we look at over here, this data is coming from the PCR. So I'm connecting with other machines now. We will be looking at the data in the real time. I will be sharing with you some of the data, and it give you information. So this is basically the current running status of that particular machines, which I tell you that we start the cycle. At current time, the machine is in real time is over here. This real time, because the initial process of cDNA synthesis starts, so nothing is appearing on the screen. It will take some time to appear on the screen. This is the remaining time, which is a countdown time, that this time is going on. Here on the real time, this is the option of plate setup. If we look at the view or edit plate setup, so now I can label this plate, I can give it the name or whatever I want. I can select the fluorophore over here, for example, in this particular color. I want to change the color of this particular dyes or the writing. So they give me the color information. You can see this color is changed now, and I select OK. The machine asked me now for making all of these changes. The machine asked me, do I want to save these changes or apply these changes? I said yes, and these changes will be applied over here. So this the plate which is currently running on the machine. I minimize this plate, and we are going for another data plate over here. This data plate I'm going to open. Yes, the machine is running, and we open this particular plate. The plate is opened. Now we can this is basically the initial screen once a PCR cycle is completed. So this type of the screen is appearing on the machine. So once this screen is appearing on the machine, we can look at the screen and we can evaluate or analyze this particular data file. So by clicking over here, it gives me all the selection of this process. Once I click over, right click over here, we go for the labeling name. So we can simple name. So all of the name which we give on this particular plate appear on this plate. So each well is basically labeled over here. If we want to look for the some of the city value of this all, so we can sort these all, and the values are appearing over here. So I just look for a particular the ENC plate. What this ENC mean? This plate basically, this particular peak over here give me the information about the IC, that's extraction control or internal control. So this peak is appearing over here. It show me that the extraction process, the RNA extraction process from that particular well is successfully completed. All the RNA is extracted from that well. And this is a negative well. It's ENC means we only put the control sample for the RNA extraction and no other sample in this particular well. While here is the EPC. It give me the information that the positive control which we extracted the COVID DNA or COVID RNA is basically extracted from this extraction process, and this give me the positive control information. While in the positive control we put the internal control or extraction control. So all of this is appearing over here. If we click it over here on this particular well, we select this particular, so it give me the information that the FAM signal, the FAM, which is basically the COVID target, the CK value is 17.63. It means that this particular sequence or this particular sample is positive for the COVID. Of course, it will be positive because in this particular case, we put the positive DNA, the viral DNA is DNA because it's not in the template. While here is the negative control. This is only the PCR master mix reactions, and water is placed in this master mix. So it gives us the amplification process, and no amplification will take place in this process. Just because there is no template is present, no positive or negative RNA is present, and no amplification will take place over here. The second information which we get from this negative control is that the all of our reagents, the master mix, the water we are using, the buffer we are using for this preparation, for this process is basically is clear or is free of all type of contaminants. There is no DNA or no RNA, no extra RNA is present in this sample. So it validates our process of this PCR amplification. It's actually a very important process for this validation process of the PCR. If these our control, these extraction control, positive control and negative control are not valid, any of the contaminations or peak are appearing over here, it means that our PCR reaction, our PCR process, our PCR batch is not valid, and we need to repeat all of this extraction process, the PCR process. While here is the positive control, this is only the PCR positive control. No extraction will be present over here because we did not put any of the extraction. So this is the positive control sample of the RNA of the process. So initially, all of these four well which we selected is basically giving us information about the controls, whether our particular PCR batch is valid or not valid. All the information coming from this four sample. Now, if we going to one of the particular one or all of the sample, and in this particular case, if another example, if I'm looking for whether there is any flaws in the extraction process, either any well is there where RNA is not successfully extracted, I just select this all, peek over here, and it give me the information that among all of these sample which are highlighted in orange color is basically positive for the internal control extraction process, except these two samples. So these two samples are basically the extraction control, the hex channel, the hex dye is basically not selected over here. If I look it over here, these three wells is basically the positive well where the viral sequence is detected in this particular well over here. While similarly in this process there is two well, these two well basically which are not positive for the internal control sequence. Over here we can see it give me the information that extraction process from these two well is not successful, and we need to repeat the extraction process from these two wells. So we immediately send these two samples again for the extraction from the RNA, and the RNA is extracted from the sample and PCR is performed. And this way our process is validated. So here again we have some more information. well, for looking for this visualizing or validating this process, we can look for one by one. This particular well is okay, this is okay, this. So, and this way we can, or we can click on this column, like one where written is, so we click it on it, and it select all the wells. So again, if I'm looking for the for this complete column, if this extraction process is valid, I select all of them and it's highlighted. Yes, the extraction process is valid. The extraction process is valid again over here. This valid over here. Similarly is positive, no positive sample is appearing over here in this particular well, while here the two of the sample are positive. These are the extraction positive control and positive control. So it give me information that these are the positive sample, basically the positive what we added the positive already, those positive. If we look at this particular well, so among this all of this batch, this F9 is basically a positive sample. This F9 is basically a positive sample. It gives me information that this particular patient is, and this particular patient, the COVID is detected. So again, if we look at the CQ value or CT value, it's 25.63. It show me that this sample is basically positive for the PCR for the COVID-19. I'm switching again toward my presentation. We discussed some of the. Okay. Well, here this is the negative control. I tell you before that this among the negative control, this is the negative peak, which is the red color is for COVID target, while the blue color is for the extraction control. So in this, the negative control, no peak will appear, and the negative control validate the PCR process, while this one is the extraction control. It means that the extraction process is valid or not. So during the batch of the automated extraction process, whether this is a 96 well machine extraction process, whether this is the 32 well machine extraction process, or this is the 24 extraction machines, 24 samples simultaneously at a single extraction time. And all of these machines, we use one of the well for extraction control, and it validates the extraction process, whether the extraction is performed is valid or not. If we look at the PCR machine, so here we are basically talk about the validation process of a particular peak which are appearing. So this one is called the baseline peak. It means that the baseline is appearing, and again this is the baseline. No fluorescence is appearing on this particular line. The baseline is extended up to a 5 cycle, no fluorescence appearing, 10 cycle, no fluorescence, 15 cycle, 20 cycle, no fluorescence. After crossing the 20 cycle, there is some exponential phase where the fluorescence is increasing. So up to a 20 cycle, it start, the PCR machine start the detection of the fluorophore at the 20 cycle, and the process of this detection is basically increasing as the cycle increase. If we look at a 25 cycle over here, so the fluorescence is increased. If we look at the 30 cycle, so it reach almost to the maximum fluorescence. So in between this phase, the linear phase where the fluorescence quantity is increases, that's RFU, the relative fluorescence unit is increasing. It gives us information that the during each cycle of the amplification, the probe is breaking down, the fluorescence is released, and these fluorescence are basically fluorescence signal is read by the optical system of the machine, and once the optical system of the machine read these fluorescence, the data is recorded and appearing on the screen. So as much as the fluorescence is exponentially increases, and a phase reach where it is a plateau. Its plateau phase means that all the FAM, that the fluorescence target the polymerases, the DNTPs molecule, and the primer sequences are utilized by this amplification process, and there is no more increase in the fluorescence. So a plateau is reached, and again this plateau is linear, and it shows the information that the process is complete. That particular well don't have any other DNTPs or any other primer molecule, the probe molecule, whatever were present over there were utilized by during this reaction. And this basically the information we get from this graph or the PCR data output. Over here, we can see the positive control. I tell you before, I show you on from the PCR data that positive control is important, and it shows information that if our primers are working finely, if our probe is working finely, if our polymerases are good, if any of this PCR batch we are performing, it is able to detect a positive sample, then the batch is valid. So in this particular, if the positive control sample is appearing, it means that yes, this particular PCR batch is valid, and we can detect on, we can report this particular batch which is run, and the batch is ready for the reporting. If positive control is not appearing or the extraction control is not appearing, so it means that the batch is invalid or that particular sample is invalid. So we can repeat these particular sample or particular batches again. Well, here look at the information over here. So it is giving us the positive and negative sample. Well, here from the machine we only selected the FAM dye, that we asked the machine to show us only the FAM color or the positive color or the one channel, and don't show us all the two color or three colors simultaneously. So the machine is not showing us two or three colors at a single time, but showing us only one color. While here in this particular case, the machine is showing us multiple color. It means that the extraction control color, the IC color or hex color, and the FAM color are appearing, and it gives us information. So here this one, the very early appearing sequence, this particular sequence is basically the positive control, while here this is the positive detected sample. If you look at the information we are getting from these positive detected sample, the sequence, the peak which is appears, the bar which is appears at the early phase, it shows us that more RNA, more viral load is present, more viral RNA is present, and age long is on the sequence. The delay appearance basically showing us that very little RNA, very little viral target is basically present in this particular sample molecule. And this again, the most of the important information, that the peak should be a properly S-shaped peak. Well, if you look at over here, the peak appearing in a properly S-shape, and there is not in the next step we will be looking, if we look at over here, so the peak is basically not appearing in a proper shape. It's not taking, and it's going up and up exponentially. Here we can take two information from this particular peak. If we look at this particular sample, two information I can collect from this. The first one is very little viral RNA is present in this particular sample. Similar with these all samples where multiple such samples are present. Why I'm selecting, why am I stating that very low viral RNA is present? And because the peak is not taking the proper shape, the S shape which is required for the evaluation of this process. If our DNA is detected and it is present over here, over there in the sample. So a peak must show all of this type of behavior, which I tell you initially a baseline must appear, then the exponential phase will appear, a linear phase will appear, and a plateau will appear. If some of the sequences are missing these information, it means that whether the probe is not working properly, or during the amplification process, once the process is amplified during the later stages of the PCR amplification, due to this heating and cooling process, the probe may be degraded. And once the probe is degraded, so the peak will be appearing. But the other thing is whether the probe may be not degraded, but the viral load may be such a low that the sample is basically low sample is detected. And this particular case, when the peak is like this, so we give the sample again for the repeat, and similar with these sample as well. And this particular cases, these sample need further confirmation. We don't report such type of the sample, but we ask these sample for further confirmation, and for further confirmation again we re-extract the whole process. The RNA is extracted, PCR is performed, and if the similar result are reproduces for one another type, then we report the sample is a positive cases. But if the similar result are not reproducible, or there is any other problem, so we sometimes ask for a resampling from the patient, that a resampling is required. Or if these samples are negative and the sample runs, so we report the sample is a negative cases. Again, this is one of the very important case I will discuss. Look at this particular case. If we look at this is a positive case where a sample is detected is a positive case. If I look at over here, the PCR peak or the CT cycle is appearing very early. Once the COVID RNA is detected very early, the extraction control is basically inhibited. Why this extraction control is not appearing, but the COVID sequence is appearing? This is because the viral RNA, the viral load is too high, that the PCR cycle detected at very early phases. And once the PCR cycle detected at very early, so in the initial phase, all of the PCR ingredients or material, these include is the primer sequence, the FAM sequence. I will not say the primer sequence in this particular cases, because the primer and FAM sequences are different for these two samples. But again, the two common ingredients which are present in this PCR reaction are basically the polymerases and DNTPs. If all the DNTPs are utilized by this COVID amplification, this COVID sequence amplification, so no DNTPs are present which are utilized for the amplification of this internal control. Though the primers for this extraction control or internal controls, and the probe for this internal controls are present, and these are not utilized at all in this particular case. So here in this particular case, if we are detecting the COVID sequence, the viral sequence, and a PCR amplification process, but the internal control is not detected, or not a proper shape is taken, is we again give this sample is a positive sample because virus is present. And this inhibition is basically, as I explained you, this I will not say inhibition, but no amplification is basically for the utilizations, because of the utilization of DNTPs and the polymerases. These all are utilized over here, so no DNTPs are present, so the polymerases cannot extend or amplify this sequence. While here again, these sample, I tell you that this type of the sample need further confirmation. If this, again, we are going for the next one is for reporting a PCR cycle that how the PCR cycle is basically reported. If we look at the software, from the software, the machine give us an option that if I want to print the whole cycle, if I want to evaluate the whole cycle, whether the cycle efficiency is okay, mm, cycle efficiency is okay, and the positive or negative cases, or I want to keep this particular process or this particular data for a record. So I need to print this particular data, and this particular data can be printed from the tool options, and from the report we click on the report, and then the machine give us a complete list of all the positive samples, the negative samples. I will show you from the live sessions on the next few minutes, because I want this PCR which we already set up to be in the process and completed. So I'm going to show you this information later on. I will talking about some of the PCR troubleshooting. Here, the extraction control sample, basically they are telling us the information about the extraction control. If we look at over here, so the multiple wells are selected over here. So it means that the wells which are selected over here, they give me some of the information basically. And here, if we look at in these particular wells, so these two particular wells is basically no successful extraction is performed from these particular two wells. We record these wells because no proper extraction from these wells, and we give it for a re-extraction process. Well, here I'm showing you some of the information with you regarding the probe breakages, the probe issue, or sometimes the false fluorescence or false positive samples are basically reported. In this particular cases, we can see that the probe breakaging issue is coming on. And why I call it the probe breakaging issues, because we did not put any of the positive samples in this particular cycle, and because no positive amplification sample or sequence is present, and beside that no amplification sequence is present, some of the fluorescence signaling appear, and this need a further confirmation. If you don't put any of the positive sample and still the probe, the detection is appearing in the FAM or particular probe or whatever probe, so we need to re-perform this PCR process, or we need to look for another batch of the probe machine, the cycle, or we need to try another kit of the probe. So why is this sampling appear? This many reasons can be over there for this probe breakaging issue. That can be not a proper shipment issue. That can be not even a proper probe preparations from the company, or that can be a very exposure to a very strong light. Can be the probe can be broken down, or simply some other phenomena or some other contamination process may appearing, so the probe may be broken down. Here again, the same breakage phenomena I'm telling you about the probe degradation. While sometimes if you add only the water molecule on it, so your water may be contaminated, and you can detect, you need to check if the water molecule is not contaminated and is properly present over there. Over there, sometimes we only extract the ENC sample. It means that the extraction control is extracted, and still that particular sequence, that particular viral sequence which were not extracted during the PCR process, and still the breakaging is appearing. So what is the home take message from this all slide is basically that the probe breakaging issues may be appearing, and that issue, for that to address this type of issues, we need to check our system properly and periodically. We should not rely on one particular kit. without validating it, without properly checking it, without periodically checking it. So we periodically check all of this system, that whatever the stock we are having, they are working finely and good. In this particular cases, we look at over here, these are the positive control cases, while here the probe degradation is coming from this particular sample where no positive sample were present, but still it give us the information of the probe degradation. If we look at on this particular sequence, so it look like that this is a completely positive case, but it's actually not the case, and this because of the probe issue. And similarly, we have some other type of the kits, and this particular kit, like the current slide we are working on, this basically have more than one target and a single PCR run, and these target are RNA speed control, which is basically the control sample. The RNA sequence is over there, which is the human sample, and it give us the extraction process and the if the extraction process is valid or not. And similarly, two of the viral genes are over there. The believer believe that the two samples are good because these two samples are for more than one target of a viral gene. If there is some mutations and these sequences, so these mutations can be detected. Similarly, if any one of the two target is basically positive, it gives you information that yes, this particular case is positive. And similarly, if you have we already discussed about that for reporting a particular batch of a PCR machine, we need to validate the control. If the positive control is okay, it means the positive control will always be appearing in a PCR amplification process. If negative control, I show you that in the negative control, no sequence should appear, while here in this particular case, you can see in the negative amplification process and the negative control, while this is the ENC process where the extraction process, and this extraction process is basically detected with the help of the hex channel, another fluorescent dye, while this one is appearing in the FAM channel, which is basically for viral target. So in this particular case, we need to re-extract the complete process. We cannot report such type of a PCR batch where the controls are not okay. So all of those type of reaction, the PCR reactions which are performed where controls are not okay, so we need to extract these samples and repeat the whole process of the PCR. While here, sometime this happened during the PCR process, the PCR amplification process. Some of the PCR is invalid. Basically, there is no flaws in the extraction process, but PCR process is well valid. And this PCR process and validity can be explained with the help of like if we are working on a magnetic bead machine, so it is always important that we do not carry any of the magnet bead with the PCR, with the template, with the RNA molecule. If any of these magnetic beads were carrying with the RNA molecule, and once we add the template and the PCR reactions, so the that will inhibit the amplification process, like currently we can see on this particular screen. So what we need to do in that particular cases, we need to centrifuge this plate, and after centrifugation, all the magnetic beads will be collected, and after the magnetic beads are collected, so we perform the PCR from the same sample again, and it's valid. So in this particular cases, like there is a lot of troubleshooting which we need to perform. We need to look at the different phenomena, different aspects of this all PCR process. We need to sort it. This is the person who are working on the PCR machine. he need to take the decision whether he need to repeat the PCR cycle or he need to repeat the extraction process. So over here on this slides, I'm showing you that sometimes the extraction process are okay, but still the PCR is not valid and we need to repeat the PCR. Like here in this particular cases, we can see that we repeated the PCR and it's still okay, the PCR, and we can report this result. Well, this one is a very important slide. Sometimes this, because during this COVID outbreak, the machines are continuously utilized, like labs where we are working 24 and 24-hour shifts, and every day we initially used to have 3 shifts a day or 24-hour work. And 7 days in a week, no break, the machines are continuously in use. So in that particular case, we need to validate our machine, whether the block, the usage is, the optical system of the machines are fine, they are working, there is no contaminations and the block of the machine from where some of the false signals are basically reported. So if we look at this particular block, this block is contaminated. If we look at over here, you can see the amplification, the signal, the fluorescent signals appears on this block where no PCR plate is placed actually. Only the optical system is reading the block, and we can see that there is some of the fluorescent molecules are basically visible. This, we can locate this particular well. Once this particular well is located, so we can see over here that the block is contaminated. So we need to clean the block. Then how we clean the block? Basically, there is 2 type of processes. We periodically perform this process. The first one is we only clean the block, and the second one we decontaminate the complete process, the complete block, and these all processes. So we once we decontaminated these block, for decontamination of these block, we take 10% bleach. We clean all the wells of the PCR block with the 10% bleach for some time, and then we wash these wells with the help of distilled water, so that all the bleach, the residues, everything is removed from the block. And for third time, we take it with 70%, we clean it with the 70% ethanol, so that all the water and everything is basically evaporated and the block is clean. Again, we run the same block again on the machine, and here you can see a clearly clean block where this RFU is basically giving you the fluorescence molecule. So you can compare this a contaminated or a dirty block, or the block which give you a fluorescence, and a clean block which give you a completely clean process, which indicate that this block is completely clean and no contamination is over there. So what is the drawback over here? If our block is contaminated, if our block is contaminated and we are still using that particular machine during this process, the disadvantage is that a false positive signals may be generated, and we can give a patient positive, but actually it's not a positive sample. So this give like a false positive cases may be given in that particular cases. So this all about the background of the block and the process of the contamination. And the second case, where we don't have any of the contamination process, and we want only to clean the block. There is no contamination, discontamination is required, but only the cleaning of the block is required. For that, one don't need to perform one don't need to perform the decontamination, like the bleach process can be skipped, and you simply can clean the block with the water, with the distilled water, with the deionized water, and with the help of molecular grade 70% ethanol. So and this way we can clean the block, and our machines can be cleared from this false fluorescence or a pseudo fluorescence molecule. Well, I tell you before that we will be going toward the machines which is running on the real time, and we can detect on positive and negative samples. So this still the cycle is running on, and here on this particular machine, so the cycle is running on. Whatever well I want to detect is basically on a real time we can see. So this cycle is still going on, still a lot of time is remaining in this cycle. So once we are able to see the cycle, I will be showing you. I told you another information about that we need to print the report of a particular molecule. If we look at over here on the software, so we can see that the software can give us the QC information, like the quality control information. What this quality control information basically means, it means that those particular cases which are positive or negative can be detected here. Here we can see that the well which are giving us information about the positive sample, like the FAM sample, FAM sequence, which are appearing over here. So look at over here, this information is coming over here. So all of these well are basically unknown, without a CK value. CK value is not present. This, like the FAM value is not present, and all of these well. It means that these well are basically the negative well. So this all information, if we click on the print report, like it's coming on the tool option, and here is the report option. If we click on this report option, the machine is asking us, here is the information. The machine is basically, and we can select, we can check whatever you want while the report the information, which type of information you want to include and your data, you can check on it. And here is the information that this particular batch is over here, all the information what we want, we can print it, we can save it. Here is the plate setup, basically the well information, like which particular sample were present and which particular well. So all of these informations can be seen over here. The organogram of the peak or the PCR plate appear over here. All the positive and the negative samples are appearing over here. So this all informations can be saved in a PDF copy or whatever, or can be a soft copy can be saved, or similarly this can be printed, and the print copy can be stored in the file for future information. So here were the information we talk about, the QC information or quality control information. Another one important point is about the efficiency of the PCR machine. If we look at over here, that the PCR efficiency, which is less than 90% and greater than 110%. So in a real-time PCR machine, the efficiency is most important. The PCR efficiency may basically be lie between 90% to 110%. If the PCR efficiency is not lying between the 90 and 110%, it gives us the information that that particular sample is basically not valid, because the PCR efficiency is not proper in that particular case. So in this way, in these particular wells, we have no well which is less than 90% or no well which is greater than 110%. It means that this particular PCR batch is a perfectly run batch, and we don't need to repeat, don't have any of the issues or problem with the PCR efficiencies. In this particular case, the process, the type of data we are recording over here, or we are looking for, we only work for the detected or not detected, if a particular patient is suffering from COVID-19 or not. So this is the aim of this diagnosis process, because it was a pandemic, a breakdown, or sample or all over is coming. So the process of PCR quantification can be performed with the similar phenomena. We can do it the CT and CK values, which give us the viral load. If particular virus is present, at which particular concentration, how much viral load is present, and these particular cases. So that can be performed with this machine. For that, we basically need to serially dilute this positive control, for which a regression curve is created. And once the regression curve is created, the machine basically give us these information automatically. But in current situation, like in the pandemic, and our lab setup where we are working, so we don't work for the quantification, because we work all the time, like continuously, to only overcome the positive and negative cases, to report the data for positive detection and negative detection. But still, I will say that for the quantification of for determining the viral load, we need to serially dilute the positive control, and we need to run it on the PCR machine. A regression curve is created, and then a particular sample can be taken, whether the positive or the negative case. And similarly, and the middle, because there will be a shortage of time, I will prefer to take some of the questions, because a lot of questions are coming on. So here is which fluorescent dye you use for fluorescence detection, and what is the mechanism of fluorophore action? Well, basically, and this particular reaction, detection process we are performing is basically it depends on the kits you are using. So the fluorescence dyes or fluorophores are coming. Again, here is the phenomena. Like, look at the particular information. I will say two important points. This is the first one is which machine you are working, whether you are selecting your detection kit on the basis of machines you already have in your laboratory, or on the basis of detection kit you already have, and you want to select a machine if somebody is going to borrow or buy a new machine, or look at the inventory of the machines. So this phenomena is like, if you have a kit, for example, and you have a 3 target on this kit, and which are 3 far lying color, like the excitation and emissions of these 3 colors are lying far apart from one another. So that depend on the machine have the ability to detect these far apart colors from one another or not. So you need to select your machine. Well, the second answer, the second phenomena is, if you already have a machine and you are looking for the detection kit and you want to buy or purchase the detection kit, so you need to look at the your machine ability, that which color your machines can select, or which particular excitation and emission wavelength your machines can select, and you can detect your you can select your diagnostic kit or the probe on basis of that. Well, and this particular case where we are working, basically we work on the hex color, the wick color, the FAM color. For one of the kit we use 2 color. These are the FAM color. FAM is basically the green color, while similarly the hex color, which appear kind of yellow color or some type of, and between the orange or red, and between that color the machine have the ability to detect it. And similarly, and another kit we were working with, were we detecting the FAM color, we detecting the rocks, and we were detecting the sci-fi color. So it depend on the machine. So I during the live sessions I tell you that you can select a multiple color or as much color as you want for your machine selection. But still the answer is that it depends on which diagnostic kit you are using, and these information are given on the leaflets of the diagnostic kit, that we need to select that particular diagnostic kit. Which type of fluorescent probe and quencher dye. This basically quencher dye is basically what the quencher mean. Basically, what is the quencher dye? The quencher dye is basically the dye. anything or any materials or any molecule which inhibit the excitation of the fluorofluor molecule is a quencher. And the probe molecule basically on the probe is like a primer sequence, exactly like a primer sequence, which bind between the left and right primer and the middle of left and right primer. And once the polymerases are basically amplifying the process, the amplification is going on. So the polymerases basically amplify. The polymerase have 5' to 3' the exonuclease activity. Once during the amplification process, the polymerases reach at the probe sequence. So the 5' to 3' end of the polymerase is basically degraded, and the fluorophore molecule, which can be a hex, which can be a FEM or sci-fi or color or any of the color, is basically degraded, and it's dissociated from the quencher. And once it's dissociated from the quencher, it means it's free. Not the fluorophore with the particular excitation and emission wavelength with a particular light, it can produce the light, and that light is detected on the photodetector system, or the machine reading system can read that particular what is the sensitivity and specificity rate of the RT-PCR machine? This is really an important question. That sensitivity and specificity of the PCR machine, it depends on the machine. The machines we are using on basically the BioRad machine, CFX96 model we are using, and similarly we have a Quant Studio 5 from the Thermoscientific. We have some other machine as well, but right now in the COVID lab we are working on these 2 units. The sensitivity of these machine is 1 copy number. So if we have only 1 molecule of a viral RNA inside our PCR tube, we can detect it. But it depends on the machine to machine. Sometimes some machines the minimum copy number is 4 or 5. Depend on the machines, basically the sensitivity of the machine, that which machine is basically we are using. In our cases, we can detect 1 copy number. And again, I will give you, we'll talk a bit extent about it. If we, for example, working with the viral molecule, how much they are diluted. Like for every 10 times dilutions of the RNA molecule, there is a difference of 3 CK value or CT value. For example, I give you one of the example. Like if we want to have a sample where the CT value is appearing on 26th cycle. If we dilute that particular sample for a 10 time dilutions, then the same, if it is appearing on the 26th cycle, on the 10 time dilution, the same sample will appear on 29 cycle. If we dilute it on the 4 time, it will appearing on 28 cycle. So every 4 time dilution, 2 CT values difference, and every 10 time dilution, 3 CT value difference is appearing. So this basically the some of the very basic which are used on the how it's possible to carry out qPCR from nasopharyngeal. Is it possible to carry out a direct qPCR from nasopharyngeal sample? If yes, which kit we can use? Yes, it is possible that you can perform without a purification of the RNA molecule, whatever in the sample, so we can perform. This is exactly the similar way people perform and the research grade process that is called is the single cell PCR machines process. And similarly, a lot of reagents kits are available. I will not prefer or advertise any particular brand of the kits which are available, but yes, this is possible, and all of the major biotechnology companies are selling this such type of kits where no purification is required. And similarly, for such type of kits where no purification is required, some type of highly specific polymerases are present, and these polymerases are basically the inhibitor tolerant polymerases. They are expensive, but they are available, and yes, that can be performed on without the purification of the viral RNA. We can perform directly from the sample. only we, is it known that coronavirus has more strain? Is the change in the primer sequence between the strain? Oh, yes, a good question is basically the strain is basically varying, and there is always variations between like the genetic shift and drift is basically taking place, and once this genetic shift and drift is taking place during the process, and in case of coronavirus cases, this is a highly mutated virus, like the shift and drift is always possible, and these coronavirus cases and mutation can be possible. So the second part, like the primer sequence, so the primer is basically, once we are designing the primers are always designed from a highly conserved region. Once we are designing the primer from a highly conserved region, it means that that particular region is not mutating it all the time, but still there is a mutation is possible. But the answer is, if there is a mutation on just a single nucleotide, the PCR primer will still amplify this process. Single mutations and the primer sequence will not inhibit the process of the PCR mutation. But the answer is that these primer are basically designed from the highly conserved region, and there is very little chances that a PCR process is inhibited or is not performed or false negative or false positive because of the primer sequence. Which one viral gene is conserved and more accurate to detect? I think I shared on the one of the slides the name of the different companies. The different kits are available. The primer sequence of particular company I will not advertise actually, but if you look at the literature, a lot of literature is available on this particular topic, and similarly CDC and WHO issue the guidelines for this information, and there is freely available on their website. Their sequence, all the information are present on their website, and one can take the sequence or the primers or whatever, or the probe sequence, and one can synthesize them. Only we can detect the virus and positive or negative. Can we also detect the viral load also, that the patient is either our initial phase or late phase? Yes, that's possible. We can detect the viral load, but I tell you before that we are working initially. Right now the load is basically because we know, like the country basically tackling the virus, the load is basically the PCR lab load is basically reduced, but initially we were working 24 hours and 7 days a week only to report the positive and negative cases. Like if we are working here on the cases like to detect the viral load, basically it will take a lot of time and a lot of more efforts. So the speed of detection or the cases we were reporting will be reduced. If we are detecting, for example, right now, if we are reporting 1500 or 1600 samples patients in a single day, if we calculate their viral loads, maybe we will be able only to report 200 or 300 cases in a day. So this is possible, but actually right now in this pandemic situation, we are not doing that. We just look for the positive or negative cases. But if somebody is looking for a research grade investigation, or some of the patient is over there and somebody is interested to look at the viral load, yes, this is possible and one can do. What are the difference between the hybridization and elongation during the PCR process? PCR step. Hybridization and elongation during the PCR process step is basically a very simple mechanism. Hybridization is when during the denaturation process, when the double-stranded DNA is denatured and double-stranded DNA is converted to a single-stranded DNA molecule. Once the temperature is reduced to 60 degrees, the primer hybridized with, make a hydrogen bonding with a particular sequence, and then the polymerizing elongated. So the process of elongation is basically polymerization. It is also called And the polymerization and hybridization is annealing of the primers with the DNA sequence is basically a hybridization. Uh, uh, some patients have all the symptoms of COVID-19 but tested, but their sample result should be negative. What is the comment about these patients? Yes, on the very first day, I think Dr. Rashid basically discussed about it, that the sensitivity of nasopharyngeal swab or oropharyngeal swab is basically 62%. So 62% of the cases will be detected positive if we take the oropharyngeal or nasopharyngeal swab, while 62% cases coming positive or negative cases, a lot of mechanism can be explained basically, but as we are taking the viral load may be very low, or not a proper sample will be collected, or virus may be not shed off during the process, so that's why they are not detected. And similarly, these are the recommendation from the WHO, the CDC, that this sensitivity of these oro and nasopharyngeal swab is basically 60%, 62%. While alternatively, there is another technique that is called is the alveolar fluid, basically, whose sensitivity is 92% for the RT-PCR. But in that particular case, it's a highly painful procedure to isolate the sample from the alveolar cavity. So basically, I would say it's not possible to isolate the sample during the biopsies. Yes, it is possible in the biopsies on some of the cases, but for every patient, it's not possible to isolate the alveolar fluid. So basically, the detection rate is very low compared to the other one. Why do you think that master mix that is prepared and the lab technician for the molecular diagnosis of the COVID-19? What do you think that master mix that is prepared by the lab technician? Or is there any drawback, and what is the concentration of different reagents of DNTB buffer, sterile water, and something like this? Master mix can be prepared by a lab technician, but I would say that the importance of trained technician during this COVID-19 is really important. And these were the technicians who deal with all of the process. I don't think so that there is any problem with the trained technicians who is performing the master mix for COVID detection. And the second thing is the second part of your question is the concentration of the DNTPs on this all this basically the similar PCR reaction, whatever we are performing normally in the laboratory grade amplification or detection system. Yes, you can perform, you can use the same one, but in the current situation where if you are using some of the prepared kit for the detections of COVID-19, so you don't need to prepare, you don't need to go for the calculation of these all processes. Basically, the manufacturer instruction, just follow them and you can go for it. While at the research grade, we say that you can start from a very low concentration of primer, it's a chronologically. Basically, you optimize it. We sometimes use a 500 nanomolar concentration of the primer and probe, and sometimes we work with a 100 nanomolar concentration of all these molecules. Similarly, for the magnesium chloride, we sometimes take 0.5 molar magnesium or manganese chloride, but sometimes we need to increase it to 2 micromolar. This all depend on the situation. Basically, if you are working on some of the novel assay which you want to establish for the COVID detection, you need to optimize all of these steps, all of these different phenomena. But in that particular case, in the COVID detection, if you are not working with optimization and working with already commercially available kit, so just go for whatever the manufacturer instruction is. Follow these instructions and report the sample. I want to know some more information about the target gene. If you want to know some more information about the target gene of the COVID detection, I would recommend to go to the CDC website. Just go over there. and you will find a lot of sequences, a lot of information, a lot of or simply just Google it. A lot of research papers are available on the COVID information. Further you want, whatever, I think we shared the manual of our with you people. We shared the manual of this workshop, and that manual as well, the target sequence are mentioned. Even you can find it over there. And my presentation, I show you the some of the comparison from one of the study where they compared the 7 different commercially available kits and their targets. Their accession number is available. You can find that accession number in NCDI, and you can retrieve these sequence as well very well and easily. So it's no problem for this all. How many genes you detect at a time in a single sample? Basically, I tell you before that we have some kits, like one of our system, we use 2 kits, two genes, basically one for the extraction control process, like the control process, while another gene for the viral RNA, while another PCR process, another kit we use RF1AB gene for viral target, and we use N gene for a viral target. Now, while there is another kit where we use 3 different genes for a viral detection, and these are the E gene, RF1AB gene, and N gene. So it depends on kit to kit. Like right now, we have one single gene target for viral target. We have 2 viral target. We have 3 viral target. This is the PCR which can be performed, and that's called is the multiplexing. Like in a single PCR reaction, you can perform a multiple target amplification. That's called the multiplexing. Yes, this multiplexing technique can be performed. How we study viral load by using regression graph. I think very difficult for me to explain to you, and this all over here. Just will simply say plot the regression curve by serially diluting the positive control RNA. Once you serially dilute the positive control RNA, plot the regression curve, and once you plot the regression curve, just ask the software, and you can easily do it with the software. And alternatively, you can also perform all of these calculation with the Excel. So with the Excel sheet, you can perform all of these calculation, and it's freely available. A lot of literature on the Google. Just Google it. This is a free available literature. You can do it. If any problem you want me to involve, yes, I'm available. You can just contact me, and we can work on it. What is the equation that converts CQ viral load to the standard curve? CQ viral load, or should I use a standard curve? CQ and CT, these are basically the terminology different companies are using. CQ is exactly similar to CT. If you want to convert this to the viral load or the expression, if you are using the similar machine, it's basically it's basically two type of the process. One is the fold change process, while another one is the normalized process. For the normalized process, whatever your value is, just divided it on the control sample value, and you will have delta CT value, and then you need to find a delta CT value. And once you have the delta CT value, you need to convert it a fold change, and once you convert it to the fold change, just plot it on the Excel sheet. And all of this calculation and the process basically is provided to you by the software, provided to you by the software. So you can go for the software and can find it easily. While here, if you look at my screen, I can share with you. Look at my screen. Over here is the gene expression. Just click it over here. Set your experimental setting. Just select your experimental setting, and whatever the experimental setting you select, you can this software, basically, if you are working with this machine, so you can easily select. Over here you have the option where you can select the regression curve, the QC, and the gene expression. All of these can be done with this software. If I think there is a lot of other questions, I should answer those questions first. And if you are interested in this all, you can privately contact me and I can explain you, can tell you all of these things on the software and on the other way. How can we identify a false negative and false positive result during the mutations? During the mutations, I will say that and that particular cases, if you say, and wherever particular global location you are were there due to your geo position. So if you already think that there is mutation in that particular region, so I will say that go for the multiple target detection. Go for a kit for not a single target kit, but for a 2 or 3 target kit. And that particular case, if you have 2 or 3 particular kits of 2 or 3 particular targets, so if there is mutation in any one of the target, so you will be able to identify the second or third target. So similarly, CQ value, what is the CQ value for positive and negative cases? Yes, the CQ value for positive and negative cases, this is really an important to share you this information. Look at over here, what we basically do in that particular cases. If in our case, if our peak is normalized, if our peak is straight, so we give it due to the peak analysis, if the peak is in a plateau position, like reach to a minimum maximum, the peak level are determined properly. What I would say, look at the position. If your peak have the baseline, exponential, linear phase, and plateau phase, so don't, if all of these phases, that is in terminology of molecular biology, that is called as, or terminology of PCR, that is called as the S-shaped peak. If your peak is S-shaped, just go for the detection, go for a positive detection of that particular case. If your peak is not S-shaped, but then you need to repeat it once again for the second time. I already, I think, shared this information that in that particular case, you need to go for repeat cases and that particular cases, and just okay, like this, fine. What can be the reason for some patients die of COVID-19 while other, including CT scan and tuberculosis positive, yet they are real-time PCR negative? Yes, this is a very important question you ask about. The PCR, the CT scan or tomography or whatever, giving you the information that the particular patient is positive. That's basically this tomographic information or the CT scan information is basically provided from your lung, and all of the phenomena which is happening in your lung is basically the immunoresponse of your body. It's not because of the virus. So the virus is basically not accumulating in the lung, and the pathologies which are created in response of the virus is basically from our own defense mechanism. That's our own immunological response of the body, is not because of the virus. So yes, of course, the immunological response is produced because of the virus, but if a patient is negative from the PCR, showing negative from the PCR, it means the virus is cleared from the body by the immunoresponses of the body, and is not because of the like there was no virus and COVID is still there. So virus may be cleared, but due to the immunoresponse of the body, so one need to come out of that particular immunoresponse of the body. That's because of the basically some of the very high inflammations in the lung, and similarly thrombosis happen in the lung. So this complication or to the ventilation or this detection, this because of the immunoresponse of the body, not because of the virus immediately. How do you interpret if the case if a peak of FEM appear near 20 cycle? You say very early, but RFU value is not so high. So how the troubleshoot this one? Yes, this is a very important question. I will look at the RFU value of my positive control. If as such a high case where what you are saying that is virus is detecting very early, I will look at the RFU value of my positive control sample. If RFU value of my positive control sample is comparable with the positive sample, I will straightforward report it a positive case, whatever is the RFU unit. If on the other hand, your sample is detected a very early positive, but there is no comparison in the RFU value. For example, your RFU value is 200, while the positive control is showing the RFU value of 2000. So still that's negative case. You need to further reconfirm it. You need to repeat the sample. Is viral infection is compared to whole? Is a viral RNA infectious? No. Viral RNA is not infectious. If somebody take a viral RNA, if we are performing a PCR reaction, I take the viral RNA, I just drink all the RNA from the virus, nothing will happen to me. So viral RNA is not infectious. Sorry. I'm again going to share the screen which is going on the live and our PCR reaction. Here is still 21 minutes remaining, and this the reaction which is going on the other PCR reaction. While here, look it over here on this particular batch, we can analyze the result. Here is these are the channel selection. If I select I remove the hex channel from over here, so it's showing me only in the positive cases. Still my PCR is going on, and I can see that this case is the positive case, that yes, this is the positive case, positive over here this one is a positive case of these two. Well, these are the positive cases. So in a similar cases, look at over there, this is the beauty of the real-time PCR machine, that still a reaction is going on, still the cycle is not completed, and I can see a clearly positive cases and this particular reaction. While just running for this reaction, similarly we can look at our control sample. If the hex is appearing, hex sample are appearing, the extraction is fine or not. So all of these information can be obtained over here. And similarly we can label this reaction on a lifetime. If I click it on it, we can label the sample over here. Just show me the sample name, and the sample name are showing, and I can locate that which particular patient is basically positive and which particular patient is negative. So all of this information is basically on a lifetime. We can see it over here. Well, just one of the very important thing I will share on this CT and CK value, while on the live screen where the cycle is going on, like right now the cycle is going on. If we look at the data for these particular, so we are not able to see the CK value over here. But on the scale bar, one can see the CK value over there. Again over there, and we can share this information over there. Just for a very good will, I will share one of the very important information again with you. That is called is the CQ determination mode. That is the single threshold mode and a regression mode. I'm on this particular screen, whatever I'm sharing over here, I'm selecting the FEM signal, and here is the baseline basically. If some of the sample, like for example, look at over here, the baseline, if I'm looking at over here, the baseline is basically appearing some of the sample. Well, this is the maximum fluorescence on the baseline, which is appearing on this particular well. So my baseline should be above this particular line. So below this fluorescence, everything is negative, and above this fluorescence, it means that this baseline means that cycle where it is going to start, the detection of that particular cycle is starting from this line, where this baseline, this sequence is intersecting the peak of a positive or a negative sample, is basically the start of the CT value, and this can be determined over here. So this machine is basically give me an advantage that it automatically calculate the baseline from the each well, and that can be determined by just over here, just click on the setting, and from setting the CQ determination mode and select the regression option. Once you selected the regression option, it means all of the data, there is no, one don't need to look for the baseline. The machine automatically detect the negative fluorescence or the fluorescence which is coming from the baseline from all of these, and we can view the data on the log sheet as well. So on the log scale, the data will appear like this, while on the linear scale, the data is appearing like this. So and this particular case, these are the positive cases. This one is the positive case over here, that is the F9. So if I select this F9 over here, this is F10, this is F9. So over here, look at, there is no baseline is appearing. If I change the setting to a single threshold mode, so the baseline is now appearing. This baseline can be varied, can be adjusted, and the technician, the worker, the one who is interpreting the data basically need to put the proper position of this baseline. But if you want to work with the automatically selecting the baseline, just select the regression mode, and you don't need to set the baseline. The machine is setting it all of itself. And similarly, this is the positive control and this extraction positive control. So look at over here, whatever the data is coming is automatically calculated. While the similar case, if the same data is taken from the single threshold, and if you are adjusting the baseline, so look at over here, this data is changing continuously by changing the baseline. If, for example, this positive control is appearing on 16th cycle, if I adjust the baseline, it is now appearing on the 17th cycle. But if I convert the same analysis for the regression mode, there is no baseline, so everything which is appear is automatically calculated. One don't need to adjust anything, everything is by default. So this is basically the live screen which is running on over here. Yes, this screen, the live screen which is running on over here, and it's 15 minutes that the cycle will be completed. For right now, why do we do not detect virus and the blood? Because the COVID virus is not a kind of viremic type of virus. In the blood sample, you only detect the viruses, the pathogens where viremia is taking place. While in this particular cases, the virus is growing on the outer epithelial membrane of this respiratory canal, is not growing inside the blood or vessels or something over here. If in a very few cases, and a very rare cases, viremia may happen, but in all of the cases, viremia may not happen. So that's why in a majority cases, but this is not recommended, that the blood sample is not recommended for the detection of the viruses, because viruses are not every viral patient, every COVID patient, the virus is not going in the blood. So viremia is not happening in the COVID-19 infection. So that's why we are not taking the viruses. Any other question? I will conclude my session over here. Thank you very much. If somebody want any further discussion or something, explanation or anything, can contact me, can write to me, and I can answer you on my behalf or whatever the discussion I'm taking off, Dr. Atiyatul Wahab will taking on the screen and will proceed the session.
Thank you so much, Dr. Irfan. Now, after 3 days of very extensive training course, we are at the final part, that is the concluding session. And first, I would like to request Muhammad Bilal, Qari Muhammad Bilal, for the recitation of the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إذا جاء نصر الله والفتح ورأيت الناس يدخلون في دين الله أفواجا فسبح بحمد ربك واستغفره إنه كان توابا صدق الله العظيم.
Jazakallah. On behalf of the organizing committee, I, Dr. Atiyatul Wahab, welcome you all in the concluding ceremony of comstech-oic CBS online certificate course entitled Diagnosis of COVID-19, Sample Collection, RNA Extraction, qPCR, and Data Interpretation. I would like to thank all the wonderful people who attend this workshop and make it successful. This workshop is one of its kind, and we have more than 300 registered participants, which represent your interest. I would also like to thank all the resource persons, including Dr. Muhammad. You can see the whole team. Dr. Muhammad Rashid, can you please wave your hand? Dr. Sabah Farooq, Dr. Hana Iqbal, Dr. Irfan Khan, and a team of volunteers, Ms. Mariam Siddiqui, Ms. Umme Habiba, Ms. Ikra Ahmed, Ms. Kiran Fida, Mr. Hafiz Bilal, Ms. Sabita, Mr. Umair, and our translator, Dr. Kasali, who has done all the French translation. Thank you so much, Dr. Kasali. Our ID staff, Mr. Shamshad, Mr. Salman, and Mr. Junaid. Thank you so much all. Now, I would like to request our beloved teacher, Professor Dr. Muhammad Iqbal Chaudhry, for the concluding remarks.
Hello, Assalamu alaikum. Can you hear me now?
Yes, sir, we can hear you.
Okay, Bismillah ar-Rahman ar-Rahim. Dear colleagues, Assalamu alaikum. I can see lots of friends and colleagues. It's such a wonderful to see all of you. And by all standards, I rate this event as absolutely world class. You know, by virtue of the number of people participated, I haven't really seen over 200 people attending any virtual meeting. And there's so much of interest. I was reading all that comments. So this has actually shown that the topic has an interest and the team has created an excellent learning module, which hopefully will benefit others. Second is that the topic itself is so extremely important. You see, it's not only COVID today, but technology of learning how to use PCR and RNAs and DNAs with so much of technical details, where so many pitfalls exist. You may really go absolutely wrong and create results which are of no worth. But Team NIV has given you an absolutely wonderful idea of what are the problems, what are the issues, how to conduct everything. Has made this event very, very successful. I also think that this event is successful because of international cooperation. You know, yesterday I was delivering a lecture in an event organized by our Institute of Peace and Development, and some very prominent ambassadors were also on board. And I said, COVID-19 pandemic is unfortunately something where we see failure of international cooperation. You know, failure of international cooperation to the limit when international cooperation was lowest ever. And countries were left, especially developing countries, left on their own to handle such a health crisis of unparalleled magnitude and its socio-economic impact. Nobody came to help each other, literally no one. And countries were left on their own to tackle a problem of an unparalleled magnitude. So I feel that this event is an excellent example where international cooperation come forth, where we can see lots of people from different countries participating in an event, actively contributing, asking questions and all that. So Team NIV developed all the appreciation. I see these brilliant people who have done an absolutely wonderful work. They are real star of today. But all of you, for your interest, because I'm sure that if they didn't have 200 people, and if they had only 20 people, they would have never been motivated to that level. You find them smiling and happy because they have talked to so many people from different countries. And I'm sure they have also benefited from this interaction. This is one of a unique event because of a reason that though we have lots of virtual meetings become a fashion of today, where we don't meet physically and we don't have face-to-face interaction. And often these meetings are spread class, you know, they just, people come and deliver whatever they wish to, and then unmute, they mute their camera and turn off their camera and mute their mic, then start playing with their grandchildren. So this has not happened here. I see everybody was so extremely attentive for 3 full days. And this is a great success. And you know, I see lots of young people, but at the same time, I see senior scientists from Indonesia, from Bangladesh, from Azerbaijan participating in this event, which actually shows that their presence was a source of strength. Because I'm sure they know a lot more than my team and NIV, but their presence really mean that they really wanted to be part of this international cooperation which Comstech has created. Now, I would say that this event has been, by all standard, beginning of a new kind of activities which we intend to do. We will certainly move forward to face-to-face meetings at some point of time also, but we would continue to have such activities which are more practical, day-to-day, and which are relevant of developing the capacity of people all around. On behalf of Comstech Ministerial Committee for 57 Muslim countries, for Science and Technology Cooperation, I would like to express my gratitude to Team National Institute of Virology and to all of you for your very active participation in this very unique and a very successful event. I'd like to thank all of you for being part of a learning exercise. Remember, learning is part of every religion. Keep learning. And this is the message, because only by learning we would be able to handle situation in future, and only by learning we'll be able to help save humanity. So best of luck. May God bless you all, and thank you very much.
Thank you so much, sir. And with this, I would like to announce that the certificates of all participants who have attended the workshop will be sent to them by email within 2 days. Thank you so much all, and Khuda Hafiz. Khuda Hafiz.